everyone, could I please, so I'm going to introduce Andrew. So he is a professor in the School of Biosciences. So he has sequenced the genome, if my research is correct, the platypus, the Tamar wallaby, as well as the thylacine, of course.
Yep.
We wouldn't have introduced or invited you to talk about the thylacine if you hadn't. And recently the Dunnart as well,
Yep.
which is fantastic. And I believe also ISO number 2, you're not loving it like the rest of us.
No, not loving it. Yeah, it's a tough one.
It is tough. All right. So to all the 61 attendees from all over the world, it sounds, which is fantastic. Could you please, we're not going to be able to hear you, but attempt to clap loud enough for us to hear. Please give a round of applause for Andrew and his lecture on cloning the thylacine.
Thank you very much. Thank you very much.
I'll leave you to it.
To do this as well. I'm really passionate about science and Science Week, and I've spoken at Science Week a few times because I absolutely love Science Week. I love getting our new generation of scientists really enthusiastic about the kind of research that can be done. So it's a great honour to be asked to speak today, and thank you all for coming along to listen as well. So today, as Julie has already told you, I'm going to be talking about our potential for de-extincting the thylacine. And I think the talks are initially called cloning the thylacine, but there is a lot of differences between cloning and de-extincting that are actually really important. So it is de-extincting, and I'll explain why that's important as I go through it and what implications that has for using this technology to actually try and bring an extinct animal back. So I'll just check, Julia, you can see my full screen fine still?
Yes, all fine.
Perfect, all right, great. So, let's see if I can change a slide. That would be helpful. Here we go. So hopefully you've all seen this footage before and you're all familiar with the thylacine, but it is one of the most enigmatic marsupial species that we had in this country. And of course, it's infamous because it is now extinct. So it's an absolute tragic tale of really very strongly human-driven extinction, that we actually hunted these amazing animals completely to extinction in Tasmania. And the last animal died in 1936, as you can see written there. And this is some footage from the Hobart Zoo of some of the captive thylacines they had around the turn of the century. And you can see them there. They really are just an incredibly unique animal. The thing that made them so unique is their shape. They look almost identical to a dog, which is a eutherian or placental mammal, but they haven't shared a common ancestor with the dogs that we all know and love for about 160 million years. So they're very, very distantly related to dogs, yet they look almost identical. Obviously, despite looking identical to a dog, they also had a pouch, obviously, because they're a marsupial, and that's where they raise their young. And they typically would have 4 babies at a time that were living in that pouch and reared by the mother in their pouch. The other thing that makes them really special is they were an apex predator. So there are those animals that sit right at the very top of the food chain and eat all the other animals beneath them. Now, if you think of our other group of mammals, the placental mammals or eutherian mammals, there are tons of examples of apex predators that you can think of. You've got foxes and wolves, you've got all the big cats, you've got bears, killer whales, dolphins. These are all examples of apex predators, those animals that sit right at the top. But now if you think about marsupial mammals, we don't have any other examples of an apex predator. This was the only marsupial that we had that lived in modern times that was an apex predator marsupial. So it was an incredibly unique animal because of all of these various aspects about it. And because of that, and it just is an incredibly, awesome looking animal, we really wanted to learn in my lab a lot more about the unique biology of this species, what made it so unique, and what can we learn even from an extinct animal? What can we do even when that animal's completely gone? Can we still use science and biology to go back and really start to understand more about the biology of this incredible living marsupial that's now extinct? So the way that my lab chose to go about this was to sequence its genome. So the genome is all of the DNA that we have that's contained within the nucleus of every single cell in our body. And that DNA contains the complete blueprint or set of instructions, if you like, that explains what it is or the instructions that you need to build that animal. So if you can sequence the entire genome of a thylacine, you do have that blueprint instruction booklet of what it is that you need to build a complete thylacine from those instructions. There's a lot that we can also get from a genome that goes beyond just those instructions. We can also use that DNA to understand more about its population size and population ecology over time. So we can actually look at the DNA and figure out how diverse its population was, how big it was, looking back in time, which is an amazing thing that we can do with a genome. And we can also use it to understand more about the evolution of that species. So we can learn more about how that species fits with other marsupials that we know that are living out there, how it fit into the ecology of the environment, and how it evolved to look so dog-like, which is something that is so uniquely interesting for the thylacine amongst all of the other marsupial species. The nice thing about the thylacine as well is, although it's extinct, it's a relatively recent extinction event. So we do have very nicely preserved museum specimens, and you can see on the slide here that this is one of the museum specimens that we use. This is actually the one that we sequenced the genome from. So this is a baby thylacine, a pup or a joey, that was taken from its mother's pouch. Its mother was killed in Tasmania when they were hunting these animals to extinction. They brought the mother in and she had 4 joeys in her pouch. And then at the time they took those joeys out and they preserved them in alcohol. And that was particularly important for us in maintaining that DNA integrity that gave us then the material that we would need to undergo such a big project of trying to sequence this entire blueprint from an extinct animal. So this is actually the one that we used to go and do that from and to get that information. You've probably heard about other extinct genome sequencing projects. There are a few that are out there. The mammoth one and the Neanderthal are probably the 2 most famous ones. And so the biggest issues that you have when you're trying to sequence the genome from an extinct animal is obviously that it's dead, that we don't have those animals around. So we don't have a fresh source of nice intact DNA to figure out that genome sequence from. But the thylacine, as I said before, it's still a relatively recent extinction event within the last 100 years. So it's quite different from when we're thinking about these other attempts to sequence extinct genomes. So Neanderthals went extinct about 40,000 years ago. The mammoth went extinct about 3,500 to 10,000 years ago, depending on what specimens they've got. And so for those, it's significantly longer than we're looking for with the thylacine. But despite that difference in time, the DNA degrades very rapidly. So the quality of DNA that you can get from a Neanderthal specimen or a mammoth specimen is actually not that different from the quality that we get out of our thylacine specimen, even though it's many, many thousand years younger. That DNA degrades really quickly. What happens though when you're a Neanderthal or you're a mammoth is obviously thousands of years ago, we didn't have museums and museum curators that were going out and beautifully collecting specimens and preserving them for us. So when these died, they died out in the environment and then their bodies have decayed. They would have been covered in bacteria and fungi. And that also breaks the DNA down even further in their genome, but it also contaminates the sample. So you're left, when you collect that tissue from a mammoth or a Neanderthal skeleton or fossil or real, whatever remains that you have, that you're getting a mixed sample of not just Neanderthal or mammoth, but a lot of bacteria and fungi and other things from the environment that are on that sample. Now, for us, we don't really have to worry about that contamination with the thylacine because, as you could see from that previous slide, they took that beautiful joey and they placed it into alcohol. So we have a relatively contamination-free sample. And that's one of the things that made this such a viable project to pursue, is that we had very, very clean DNA. As I said before, it's still very highly damaged. So it gets broken up into lots and lots of tiny little pieces. So we call that fragmented. So the DNA is broken up into lots and lots of little sections. Our DNA in our own cells, and it's about the same in a thylacine, is about 3 billion bases or letter codes long. So that's the length of our genome. It takes up about 3 meters of DNA if you were to stretch the entire strand out. So it's an incredibly big molecule that's packed down into each of those cell nuclei. And so what we find is in these samples that DNA, that 3 billion letter code, is actually broken up into fragments mostly below stretches of about 100 bases or 100 letters in each little fragment. So it's an enormous job to try and sequence through that genome and put those pieces of the puzzle back together, which I'll come to in the next couple of slides. So it makes it very, very challenging for us to sequence and rebuild these extinct genomes. So we tried to get DNA from a lot of different specimens. That's what this slide's showing here. We tried initially from skins. There are hundreds of thylacine skins in museum collections all over the world. But that wasn't very good. That was really broken down. Most of the fragments, so this is showing these black blobs down here, is the DNA that we could get from these pelt samples. And most of it's at even less than that. So about 50 letters long or less. So less than 50 bits of information in each one of those little fragments. So that's simply too small for us to think about building that genome. But when we used these alcohol fixed pups, we were able to get some larger fragments of DNA that gave us the ability to build that genome. So the key thing with the thylacine is because they were preserved in these museum collections, we have a lot of very clean DNA, so it's not contaminated, but it is very highly degraded. So we say it's abundant, but degraded, broken up into lots of tiny little pieces. But that does still give us the ability to go ahead and sequence each genome. So we extracted all those broken up tiny little pieces. We got rid of the very, very smallest ones, but kept everything that was bigger than 100 letters long. And then we sequenced that genome. We actually generated around 120 billion letters of code, or billion bases. And that's about 40 times sequencing through the genome. So whenever we try and do one of these genomes, we can't just sequence through it once and try and put the pieces back together. So we effectively sequenced about 40 times more than you would need, so that we have a lot of coverage of that DNA. Every bit of DNA in the thylacine genome's hopefully been read through at least 40 times. And that gives us a lot of confidence when we're trying to rebuild that and put it back together again to build that blueprint that we need to understand more about the thylacine. Just to give you some idea of the complexities of putting those pieces back together again. So as I said before, the genome size overall is about 3 billion base pairs in length. Now, the biggest problem that we have as mammals, and marsupials are also mammals, is that about half of our genome is made up entirely of repeat sequences. And that's the same looking piece of sequence that can be just repeated over and over and over again for thousands, tens of thousands, even millions of letters or bases of our genome code. Obviously, that becomes really challenging to understand how big those regions are if our entire genome is broken up into lots of tiny little pieces. So what I liken it to is that you're trying to build a puzzle, which is what I've got depicted here, and at least half that puzzle is just blue sky, right? That's all our repeat sequences. They all look the same. We know some bits of the puzzle fit together better than others, but it's really very difficult to tell, do we have 10 bits of blue sky here, 50 bits, 100 bits? How do they fit together, and how big are those blocks of blue sky on our puzzle? It becomes extremely challenging to try and build those pieces. This is very important when we start to talk about the de-extinction of the thylacine, which I'll come to in a few slides' time, because this is where there is a lot of uncertainty in the genomes that we can build from extinct species, is particularly within this blue sky region. Now, initially we thought that these repeats, these blue sky bits, didn't really do much in terms of the biology of the animal. They weren't very important. But the more we manipulate them in living animals, the more we find out that there actually is a lot of information still that's contained within those repeats that may actually be important for some of the biology of that animal. So that's a really important thing to consider as we move forward. The other problem we have with the thylacine genome, obviously, is we have all our pieces of the puzzle, but we don't have a picture on the box. So even for the bits that aren't blue sky, we're still trying to put one of those puzzles together without the picture. So for any of you that have ever bought one of those hellish puzzles where you can buy the ones that don't have a picture on the box, you'll know just how challenging this is. It's an enormous effort to sit there and try and sift through all these pieces and figure out which bit connects to which one and which one has the best fit. So this slide has some statistics on it about the genome, which none of it's really important for this talk at all, other than to say that what you can do, though, is you can use the picture on the box from a closely related animal. So although we don't have the picture for the thylacine genome, so we don't know what it's supposed to look like, we can use that picture from a genome of another animal that's been sequenced to look at where we think all those pieces of the puzzle should go. So in the case of the thylacine, we use the Tasmanian devil genome. That was the marsupial species which had the best genome, that was the most closely related to the thylacine. There are some other species we now know from figuring out its genome that are actually more closely related to the thylacine. But at the time, this was the best that we had, was the devil genome. And so we used the devil genome as our picture on the box to try and figure out where all those pieces fit. And at the end of the day, once we had all of our fragments of the puzzle and we use big supercomputers to go and put all these pieces back together, we actually ended up with an incredibly good genome build for the thylacine. Even still to date, so we did this a couple of years ago now, it is still the best extinct genome that we have sequenced to date, in terms of it being the most complete and the best built genome of any extinct animal. So that's really good, and it certainly gives us a lot of hope when we start to discuss the prospects for de-extinction for this particular species. So there's a lot of other things that we can do with the genome, as I alluded to in the beginning. So one of them is figure out the phylogeny, or where this animal fits into the evolutionary tree of all the other marsupials. For this talk, the most important thing for this is that we found out that the thylacine is actually most closely related to the numbat. So that's its most closely related living animal. And so as we start to talk about the de-extinction side of things, this would be sort of the picture on the box, if you like, that you want to start with when you're trying to rebuild the thylacine genome. You can see here it's a lot more distantly related to the Tasmanian devil, and there's a bunch of other dasyurid marsupials that you can see up here. This is another one I'll speak a little bit about as we go through the talk, which is called the Dunnart. So this is one that's very useful for us for developing a lot of the technologies that we will need to try and bring the thylacine back. Oops. But the numbat is the one that's most close related. So now we're sequencing the numbat genome so that we can use that as the picture on the box to do an even better build of the thylacine genome to give us a much better blueprint for then moving forward with some of the de-extinction side of things. Another thing that we can do that's really cool with genomes is you can actually look at the population size of an animal over time, just from looking at the chromosomes of one animal, one individual, which is actually just a mind-blowing thing that we can do with science now, is actually take that information. Again, very complicated, big supercomputers can run these complex algorithms to analyze the genome and figure out how big the population size was of the thylacine going right back 100,000 years, which is what you can see where these lines start back here, is about 100,000 years ago. What was really interesting about this is we trace its population size over time. So this is 100,000 years ago, and on the very left, you've got modern day over here, is we can see for the thylacine, and we also did this for the Tasmanian devil, just to see what was going on with it. For both of these species, we see that they were actually quite abundant in Australia about 100,000 years ago, and then their populations started to decline. They started to decline and they continued to decline after the arrival of Aboriginal Australians. This is about 50,000 years ago. And then their populations were quite small and they had quite limited genetic health. So that meant the population itself wasn't that healthy. And this happened before Tasmania became isolated from the mainland. So we thought that animals like the Tasmanian tiger and the Tasmanian devil probably struggled to keep being a competitive species on Tasmania after the island became isolated, because you end up with a small number of individuals on that island, and that's not great for your population health. But what this experiment actually showed us was that those animals had actually declined in number and declined in population health for a long time before they became actually isolated in Tasmania. So that was sort of a very unexpected finding that we can pull out from the genome, which was really valuable for us in trying to understand more about that species, and particularly what we might need to do to help that animal out if we ever decide to bring them back from extinction. So moving into the topic that I'm sure everybody wants to hear about is, what is the reality of trying to bring an extinct animal back? So the main thing I would like to impress upon all of you here today is that it definitely is no longer science fiction, but de-extinction is really science fact. We have all of the tools and the technology that we need to bring an extinct animal back. It's not a question of if we're ever going to do it, but when it's going to happen and who will be the first one to bring a species back. The project that is moving forward the fastest at the moment is the de-extinction of the mammoth, which is the one you can see pictured down there on the National Geographic cover. And I'll explain why that's such a great poster child for de-extinction and one of the good ones to try and do this for. But the first thing that you need if you want to de-extinct a species is a really good genome sequence. So we're very fortunate that we absolutely do have that now for the thylacine. That is in hand. You also need to have a close living relative. And for us, we've now identified that probably is the numbat is going to be the best close living relative of that species. And you need that to help with the de-extinction process. And then finally, you need to be able to do assisted reproductive technologies. So that's things like IVF, where you can create an embryo and transfer an embryo into a pregnant mother to rear that baby in the species that you want to use as a surrogate to bring your animal back. And that's something that's never been achieved in marsupials before. And something that my lab is working quite a lot on at the moment is trying to get that technology up and running for marsupials. Not necessarily for a thylacine de-extinction, but more for all of the conservation potential that is out there if we can get this technology up and running for marsupials so that we can help protect those species right now. So I'm sure when everybody starts talking about de-extinction, we immediately go to Jurassic Park. So just to dispel any worries that people may have out there, the half-life of DNA is about 500 years. So in 500 years, approximately half of the DNA in your sample is completely gone. So by 7 million years back in time, for any species that has a genome about 3 gigabases, 3 billion base pairs like ours, basically all the DNA is destroyed by 7 million years. So dinosaurs went extinct about 60 million years ago. So despite what we learned in Jurassic Park, there is no dino DNA in fossilized amber with mosquitoes in it. So we're not going to be bringing back T-Rex anytime soon. And yeah, we won't see small children getting eaten by rogue dinosaurs in the near future. But the technology that they spoke about for how you would go about de-extinction, I don't know, for those of you who remember the movie, this little cartoon of Mr. DNA comes along and he tells the story of how they're going to bring the animal back. It was surprisingly accurate for what we will actually be doing when you try and de-extinct an animal. And so this is how de-extinction differs from cloning and why the two are not the same. So for cloning, and we think of Dolly the sheep that you can see in the top right there, the first thing that you need to clone an animal is a living cell. That is the absolute ground state zero that you need to then create a whole nother living individual from that one cell. Obviously, with an extinct animal, we do not have any living cells. So that's the problem. So if we want to de-extinct something, we still have to start with a living cell. We still can't create life. We can't create a living cell from just basic components. We have to start off with that living cell. So what you do is you get a living cell from your closely related living relative to your extinct species. So in the case of the mammoth that you can see up here, the closely living related species is the Asian elephant. So you start off with your Asian elephant cell, you sequence your Asian elephant genome, you sequence your woolly mammoth genome, and you look for everywhere in that genome where those two are different. So if the elephant DNA differs from the mammoth DNA, you can then go into your elephant cell and you can engineer in that change so that now the elephant DNA actually has the mammoth code in that place. So the reason that the mammoth is such a good poster child for de-extinction is it's very closely related to Asian elephants. Their genomes are extremely similar, so there's not a lot of changes that you need to make to your Asian elephant cell to actually make it a mammoth-like cell. The only thing that we can't really ever be sure of is that blue sky repeat sequence, how similar or different that is between those two species, because we're not very good at putting those bits of the puzzle together. But whether that will mean that you don't really end up with a mammoth at the end or not remains to be seen. So they're still moving ahead with that, even though some of those gaps will be there. So in terms of the thylacine, how close are we to bringing this species back? What tools do we have in place? So as I said before, the first one is sequencing the genome. That's a huge task, and it's something that we have completed and we have done very well for the thylacine. So that's really great. The second thing is that you need to edit in all of those edits into your surrogate living species. So this is where for the Asian elephant and the mammoth, they've come a long way. So they're a good chunk of the way into making all of those mammoth edits into Asian elephant cells. We haven't begun this yet for the thylacine, and that's because the thylacine and the numbat, although they're the most two closely related marsupials, they're a lot more distantly related than the Asian elephant is to the mammoth. And so because they're much more distantly related, there's a lot more changes between the thylacine genome and the numbat genome that would have to be edited. And so we need that technology to have another leap in how efficiently we can start to make those edits before we would start to consider it for the amount of changes that we need to make to our numbat cell to make them look like a thylacine. The next bit that you need to do is, once you can recreate that and can recreate a little thylacine, is have somewhere to put it. So this becomes a big problem for the woolly mammoth project, is there really isn't their habitat out there still in existence. This is an enormous animal. You can't, you know, just let them run in your local neighbourhood park and hope for the best. They really need somewhere that they can fit back into the ecological niche where they once belonged. This is the great thing for us, though, with the thylacine, is because they're such a recent de-extinction event, their whole ecological niche still exists in Tasmania, as does the entire food chain that they need to feed off. And in fact, there's a lot of evidence to suggest that having an apex predator put back at the top of that food chain can really help with a lot of the problems that we're seeing currently in Tasmania. So one of them is the devil facial tumour virus. So that's the terrible tumour that the Tasmanian devils get on their face, which has nearly wiped out the entire population. When you have an apex predator like the thylacine in that ecosystem, they go out and they eat all those sick animals. So before those animals will pass on a lot of their diseases, if they're a weaker animal in the pack, then the thylacine would go in there and it would eat those animals, and it helps control the spread of diseases like that throughout the population. So they actually really do help stabilize those ecosystems. So there's a very good reason for doing it. And the last thing is, based on that population life history curve that I showed you, we know that the thylacine wasn't in great genetic health. So we would have to also think about not just bringing them back, but also engineering in enough diversity into their population to make them a very viable animal for surviving long-term in that environment. So the only bit that we haven't really done is being able to figure out this middle step here, which is really being able to have a good surrogate species to bring them back in, and to have all of that technology for doing the gene editing and stuff worked up in marsupials. So that's what we're concentrating on at the moment. And as I said before, we're doing it using this little guy here, which is called the fat-tailed dunnart. They're about the same size as a mouse, and they're really good for breeding in captivity. They just breed, they have tons of babies. So you can see the picture down the bottom here is a pouch full of little baby dunnarts. Here's what they look like on the day of birth. They lay just 100 milligrams. That's how much they weigh when they're born. They're about the size of a grain of rice, and they're still able to crawl into mum's pouch at that age and then connect to the teat and start drinking milk. Really phenomenal animals. And by using this animal, we're able to start developing some of these technologies that we need to think about this de-extinction process. So what we've been doing with the dunnart is starting off with deriving some cell types that we can use to create whole embryos from. So with the dunnart, we started to generate induced pluripotent stem cells, or IPS cells. These cells you can generate a whole embryo from, but they're a very useful cell type to have to start doing some of this genetic engineering of putting thylacine changes into a particular genome. So we've got those cells, we've started doing some of the editing, so we can develop the tools and the techniques that we need to start to put those edits in. But as I said before, we can't think right now about doing an entire genome's worth of edits, because it simply would take several lifetimes to do that many editing with the current technology that we have. But that technology is moving forward incredibly rapidly all the time. So it's no doubt in the next 10 years, I'm sure that we will have technology that makes this far, far more feasible to think about. The great thing about having these induced pluripotent stem cells in hand now is that it's the first time that we really start to have these tools available for marsupials. And as I mentioned before, these can be incredibly useful tools for not just de-extincting marsupials, but also for conservation efforts for many of our threatened marsupial species that are right on the brink of extinction right now. So if you can start to edit the DNA of these marsupials, we can do things like engineer in better immune health to some of these very restricted populations. We can think about engineering in things like resistance to that devil facial tumour virus. And the project that we're working on in my lab at the moment is trying to engineer in cane toad toxin resistance to quolls. So these are quoll over on the right-hand side here. You can see these beautiful animals. Unfortunately, they eat cane toads. Cane toads are toxic and they die. But animals that grew up with cane toads, so cane toads, I should say, were introduced into Australia. And so our native species have not evolved alongside cane toads, and so they find their toxin particularly toxic and it does kill them. But animals that have evolved alongside cane toads, such as hedgehogs that you can see pictured here, have also evolved resistance to these toxins. And the resistance is conferred by or controlled by just a very few changes to a couple of genes in their genome. So you only need to edit a tiny, tiny amount of DNA to make our quoll resistant to cane toad toxin. So that's what we're working on at the moment, is actually trying to use our technology from the Dunarts to see if we can start to make these marsupial edits and see if we can do things like engineering resistance to that species. This would be an amazing outcome if we could, because not only then would the quolls not go extinct, but they would also then help control the spread of cane toads by actually eating this problem species. So it'd be a really great thing if we can achieve it, because we can have that 1-2 punch of saving the species and getting them to help control the ecological disaster that is cane toads. All right, so just to wrap up today's presentation, we do have the best extinct genome build that we have to date. So we've got an excellent blueprint in place to build our thylacine. I think this research really shows the value of museum collections too, that they're not just, you know, a great repository for some of these species that we can go and look at, but we can actually start to unpack and learn so much more about the biology of these animals by sequencing the genomes from some of these extinct animals where we've still got great museum collections available for them. And as I've shown before, we were able to clarify where it sits in that evolutionary tree, which is very helpful when we think about de-extinction, because we know we need the numbat for that process. We can learn about its population life history, which told us it was in poor genetic health, so we would need to engineer in some diversity into its population. And we can also learn about how it evolved as well, which I haven't really had time to speak about today, but that's another thing that we can answer from looking at its genome. It's a great candidate for de-extinction once we have the technology in place and it becomes cheaper. So we have the technology, but once it becomes cheaper to do it en masse, it really is a good one because, as I said, its ecological niche is still right there. So you could just recreate it, pop it back into Tasmania, and it should be able to live its normal life. And in the process of developing the tools for de-extinction, like these stem cells, like learning how to do marsupial gene editing, we are developing a whole suite of techniques which are really helpful for preserving some of the diversity that we've got in our marsupial populations out there right now, and also for engineering in some of these things like resistance to certain toxins to really help those populations that are right on the brink of extinction. So it can have a lot of benefit for current marsupial populations as well. I'd like to end on this. So obviously there's always the ethical question of, sure, we can bring a thylacine back, but should we bring the thylacine back? And I really like this quote from Mike Archer, who was head of the Australian Museum at the time when he said it. And I think it really resonates well with me when he said, we played God when we exterminated the thylacine. And I'd like to think we're playing the part of smart humans by trying to bring them back. And I think that's very much how I feel about these projects, is that we really did completely hunt and annihilate these species, wiping it off the face of the earth. And I feel if we do have the technology to bring back an actual thylacine, and it is there, that we owe it to this species to try and bring it back. So with that, I'll thank all my collaborators and end there. I don't think I've gone too far over time, and open it up for questions.
Thank you so much, Andrew. That was absolutely fascinating. I think everyone in the audience would no doubt agree. It was you're so articulate and so passionate about this, and it's so fantastic to hear it and to hear it so succinctly. I think using the analogy of the puzzles is probably a great analogy at any time, but I can say for myself in ISO, I've done many a puzzle, and the blue sky is the death of me, and so I can understand the uphill battle and the challenge that your work has been right now. So so thank you, and I guess well done so far, and all this amazing work that you've done. It's fantastic. Great. So yeah, so a reminder to everyone, please add any questions that you would like into the Q&A function just at the bottom of the screen. We do have some coming in already, which is fantastic. The questions, so there's a few different sort of areas of the questioning. Oh yes, great. In terms of, I guess, a bit of the technology side of things, I guess there's some ethics questions, which you did touch on towards the end as well. And I quite a few actually about sort of what would happen when, not if, when they can be de-extinct. That's sort of what the environment might throw at them at that. So let's get started. I was going to start with the Jurassic Park question because that's always been on the front of my mind, and it has been since I heard that you're going to speak, but I think you did cover that quite well, although I probably will be going back and watching that movie again, knowing that there is some scientific rigor behind it.
There is,
Good to hear. I mean, it's good to hear. Good work, researchers. Yes. So, what about the technology? So, from Zachariah, so can you resequence using the nanopore? Have you got that one? So, nanopore technology uses long
so this
reads to cover
nanopore
the gaps.
technology, it lets you read very long fragments of DNA. So it helps you with that blue sky, because rather than reading little fragments, you can get a big chunk. The problem for us with that is that we don't have big chunks. It's only little pieces. So we have tried nanopore and we use it and we get some bits, but mostly because they're all really short, it doesn't really have any advantage for us of using that technology. So it's a shame. We would love to, but we really need those big fragments of DNA in order to use it the best. Yeah.
Okay, great. So I guess following on from the technology, you're talking about, so I'm just going to add one in here now myself, because you said, you're waiting for sort of, the technology to be cheaper. Is that really the next thing that you're waiting on? Like what is the next sort of progress at the moment?
Even maybe 5 years ago, this was unthinkable that you could even attempt it with the mammoth, right? Because of the number of changes. But the technology has come so far in that time frame that now, engineering in all the mammoth changes to an Asian elephant genome has become easily feasible. Like, that's not such an insurmountable task. We're another order of magnitude again of changes, of edits that you need to make.
Yeah.
So who knows, another 5-10 years, I reckon we'll be in that ballpark of where we will be able to do this. Yeah. But it is, that DNA editing technology has just made an enormous leap in the efficiency of which we can do it and how reproducible it is. And so that was really sort of the major advance that made de-extinction suddenly become a very possible, tangible thing.
It's amazing. It's very exciting. Okay, moving on to a few, I guess you did touch on sort of the ethics of it. Is this something, obviously you did touch on, is this something that you have to, question or answer?
Yeah, it's a great question. I think what they're asking as well is, does it make us more complacent about the species that are on the brink of extinction now, right? If we go like, oh, well, we can just bring it back, who cares? So I think the main thing about de-extinction is that it's never going to be 100% thylacine because of that blue sky that we'll never be able to fill in completely faithfully. And so the ultimate goal is always, you have to preserve what we have right now. Creating those stem cells is one way that you can do that. So making those stem cells now for marsupials, we're aiming to try and collect as many marsupials as we can and make stem cells from them. So we've got a proper, faithful, cell that you could reproduce those complete animals from. So really do a cloning of those particular animals. But for de-extinction, that's part of the controversy of bringing them back as well, is what are you actually bringing back? It's always going to be some sort of hybrid between a thylacine and a numbat, or a woolly mammoth and an Asian elephant. So it remains to be seen how much they behave and act like the original species. And in many cases, we don't have anything to go by, right, to really know whether they are normal or not. We're just sort of hoping for the best. So it is still there's a lot of good reason to have a lot of, ethical discussions around doing this. And certainly at the moment, because the technology is still quite expensive, you would never want to invest money on bringing the thylacine back as opposed to using that money now to protect those so many marsupial species on the brink of extinction. So I think, as this becomes more tractable and cheaper, that's when you would go, okay, now we can have a crack at trying to bring it back.
Yeah, makes sense. Fantastic. Okay, so I think moving into a few more questions about, the when it might happen and sort of what we can put in place. So there's from Anonymous. So will they be able to adjust to today's climate and access to food plant sources?
Yeah, the thylacine, no problem. The woolly mammoth, who knows, right? So I guess that remains to be seen. So for them, that would be much more challenging. Thylacine, it hasn't been that long, so there hasn't been enough climate change or change to the species that they eat to really cause many issues for that. So I think, that would be probably fine.
And so another question. So the question is, what will the effect be on other animals? Is there the possibility of the environment and ecology becoming unstable because of their reintroduction? Is that something you're working with other? So as I said before,
I think there's good evidence to suggest that apex predators help stabilize that food chain. So they actually would do good rather than bad. But still, this is something you would do a very controlled release of, like you do with any animal sort of, rescue program, where initially you would create some and you would release them into a very large enclosure and just make sure that they're not really tipping the balance off before you would release them, across the whole of Tasmania or something like that. But they're really important questions to ask. And if somebody goes down this road of de-extincting any one of these species, it would be very carefully controlled how you would go about that next step process to make sure you don't cause a new ecological disaster by putting them back in again. And again, for the thylacine, you know, it's like 86 years or whatever since they went extinct. So again, it's not that big of an issue, but for a mammoth, what impact it's going to have on today's ecosystems, because it's never existed with them for, you know, thousands of years. So it's a much bigger question. Yeah.
Yeah, absolutely. Chris Cook, so what would be the smallest amount of genetic DNA information you would need to differentiate between individuals? Good question, Chris. And also to be able to use that information as a conservation tool.
Yeah, so I mean, they are excellent questions, and the answer is we just don't know. So we're still figuring all that stuff out, but they are excellent questions. We do know a lot more about things like their immune system now from a whole bunch of marsupials. So there's other groups that work on that and have characterized that very well. And so I think we could be pretty good at engineering in those differences to give them a very good immune system. But there are lots of other changes in our genome in other places that are also important for our genetic health. And so ideally, we would want to then go back and try and get, you know, as many specimens from skins and other things that we can get for the thylacine. And although that DNA is highly damaged, once we've got a good picture on the puzzle box, it makes it much easier to put those pieces back in. So we can have a go at sequencing some of those much more fragmented samples, just so that we can learn just sort of what diversity was out there in the population and then engineer those changes in. That would be the best thing that we could do.
Yeah. Okay, great. Now we've got maybe time for just one or two other questions. We'll just go from the top. So is there a danger, so for Michelle, is there a danger in using just a single individual's genome to de-extinct the
Yeah, so that's
thylacine?
exactly what we were just talking about. So absolutely, that is a problem. So but you know, if we can engineering all the differences between a numbat and a thylacine, engineering in enough diversity is really, like, that is child's play compared to everything else that would have to be done to get that whole genome intact. So once we are able to do it on that scale really efficiently, engineering in that diversity, I think is, you know, we'll be able to surmount that quite easily.
Yeah, okay, fantastic. All right, now we don't have much time left. I might just throw in one final one just to cap things off. So you're saying it's not a matter of if, but a matter of when. Do you think, so you said 5 to 10 years, there might be another advance in technology. Are you at the stage you think you'll be patting a thylacine in the next 10 years, 20 years, lifetime?
Yeah, I would really like to. So my fingers are crossed. I think we'll all see a woolly mammoth born in the next decade. So I think that's definitely moving forward. And as long as that technology keeps going, then I love the thylacine. And I do think that we really owe it to that species. If it becomes feasible to do it, then I will have a crack at doing it. So I certainly hope so.
Fantastic. Oh, I bet you will. Fantastic. Well, thank you so much for taking the time out to present all your fantastic work, to sharing all the work that you've done so far. And yeah, absolutely good luck with the future, and hopefully some more successes do come your way.